Thanks, Susan, and thanks everyone for your attention this afternoon. We have Nick Bertos, who's going to be telling us about bio and data banking here at the RI, for which he has a lot of experience in it, both in general and specifically at the RI since 2017, when he's been leading this biobank technology platform. If you're already interested and curious about biobanking, that's great. If you're not, I can attest that it's actually extremely valuable. Some of the most prolific research output, research programs, and researchers have really made their careers by building these types of biobanks, which allow them to be incredibly, I guess, quick in addressing cutting edge hypotheses, because they have this data stored, especially when it's well phenotyped, well curated. Really, it takes a good amount of investment up front, but then you really can reap the rewards. So I'll hand it over to Nick to tell us how things work here and where they're going. Thanks.
Okay, so thank you, Dr. Smith, for that wonderful introduction. So without further ado, I'll launch into the presentation, and I'll try to outline the opportunities, the advantages, and the potential pitfalls, or things where you do not want to be thinking to yourself a year after you started, oh, if I'd only thought of that. Moving forward here, hopefully. Okay, so I apologize in advance to anyone who's got their menu bar at the top or at the bottom. You may lose part of the slides. I suggest actually having your menu bar at the bottom. You won't lose the titles. So first of all, the definition of a biobank is that it is a collection of tissue and fluid samples or biological materials. But of course, this can include other types of samples as well, and personal health information or data. It's stored as an organized collection of searchable samples and for the purpose of conducting specific or as of yet unspecified research. Now that sounds kind of legalistic, to be honest. So the actual purpose, you're trying to store samples. Generally, I'm going to be talking about biobanks that hold specimens from human participants. There are also biobanks for various animal models, for example, but those have very different regulatory frameworks associated with them. And in the case of samples from humans, associated data, which includes the relevant clinical information, both initial and follow-up, if applicable. The consent forms that the participants sign to allow you to actually use their samples, which include there often are different versions as time goes by, and there are, of course, limitations that can be either version dependent or specific individuals who have consented to be within the biobank may have consented to more or less widespread use of their samples. Information about the samples themselves, how they were processed, where they are, if any quality control has been done, associated SOPs for processing and storage, since these, especially if you're looking at long-term biobanks, can change over time. Just to note that samples without associated data are of limited value. There is a fair number of so-called biobanks that are sort of historical. You know, a bunch of tubes that are in the back of somebody's freezer labeled with, you know, these are fibroblasts from MS patients. But nobody knows who the patients were. Nobody knows the details of their condition. In that case, the value of that set of samples for research is somewhat debatable. And one thing to keep in mind is that a biobank is a resource for research. It is not a research project in itself. There are defined studies or projects which use the resources that are held in the biobank, but the biobank itself is an independent unit. So why bank or use human samples or data when there's lots of animal models out there, there's lots of tissue culture models out there? These are very useful for mechanistic studies because they allow a lot of simultaneous parallel manipulations to be done, and in a controlled setting, but they don't recapitulate the heterogeneity that exists in human disease. And if you're using what I call a non-primary model, you're limited in looking at the features that are present in that model. You won't, if it's not in your cell line, it's not something you're going to be able to look at or discover. So they're not as useful in a discovery setting. Also for validation, a lot of times, if you discover something in T47D cells, that's great. You'd like to actually see whether or not this applies only in one individual, in a very small subset of individuals, or whether it's in fact more a global mechanism that one could look at. So it's usually for validation, requires testing of larger cohorts and real world cohorts. And from a purely practical point of view, this tends to elevate the relevance of findings and makes it a lot easier to get a grant or publish in a higher impact journal if you can show that, in fact, it is relevant to human health. So the next part is what to take into account when creating a biobank, so starting essentially from the beginning. So step 0.1 is to define what's your participant population. So which patients or which potential participants would you want to include in your biobank? This becomes very important because you can either wind up with a very wide spectrum of patients and you wind up doing a lot of collecting that in the end is not useful for your research project, or you could wind up over specializing and then realizing that you really needed to have relaxed your criteria a bit more. The problem is that it's very difficult to go back 5 years later and say, oh, I should have included those people. Well, you can only work forward and not backward. Other things to keep in mind is you need to look at which control population you might need. Do you want to be age matched, gender matched? Are there other factors you would need to look at for your controls? Is your selected population balanced for age, sex, ethnicity? And kind of an odd point, but is any group unintentionally being excluded? And I'll use as an example for that. If you collect samples from dialysis patients and you only cover the patients who come for dialysis in the morning and the afternoon, and you don't do the evening set, you're going to bias your group towards a less healthy, older, non-working population, because the group of patients on dialysis who are in a functional condition and capable of actually going to work are the ones who come in the evening sessions. So if you skip those, you've just unintentionally biased your population to be non-representative of the actual clinical group you want to look at. The other thing you need to keep in mind up front is, will you keep links to patient identity? You can choose to collect either de-identified data, so you get the sample, you get the consent, and then you break the link between the code on the sample and the name of the patient, so you no longer know who, sample XYZ003, you have no idea who it is. This makes things easier from the point of view of confidentiality, because you only need to keep the patient consent forms in one place. You don't need to be using this information for follow-up. However, it means, of course, that you don't have any follow-up. And note that if you do follow up and you keep personally identifiable data, it must be stored in a protected and secure environment, i.e. in a locked room with restricted access, or if it's out there on the net, it should be behind the MUHC firewall. And an important point to keep in mind is that samples should be coded at the biobank level using a code that is not linked to patient identity. So no MRNs, no patient initials, no telephone numbers. And that if you use it in a study that is going to be reported publicly, there should be a secondary coding system, double coding, and you shouldn't keep all the coding correlations in one place. So identifier to primary code in one place, primary code to the double code in a different place. This is important to maintain patient confidentiality. The other thing to keep in mind is you need to define what sample types you want to collect. Ideally, The simplest way is to find sample types that are already being collected for clinical needs, because you can then combine the collection with a collection in the context of clinical care. If you need to get a separate collection that adds to the expense and the logistics and the complexity of your bank, you also need to know whether you should identify specific elements and separate them up front. Thinking, for example, in the oncology setting, if you're getting patient tumors, do you want to collect the tumor core, the tumor margin, and the stroma separately, or do you want to collect just tumor and normal, or just tumor? Because it's very hard to go back afterwards and re-separate those. This is something that needs to be worked out up front. Yeah. Another question is, do you want to do longitudinal sampling? Do you want to get urine samples every 3 weeks from patients? And again, it's good to look at what the clinical sort of treatment protocols for patients in that setting are, because if they come back for a urine sample every 4 weeks, it's probably a good idea to combine the clinical sample collection with the sample collection for research use, just both for logistics and for the convenience of the patient as well. And the point of making sure that you identify exactly what you want up front is, unlike in mice, where you can always say, oh, well, I didn't get the 14-day time points. Oh, but I'll just, we've got another batch of those mice, so we can just repeat the experiment with those. Humans, you can't go back and repeat the experiment. They're far too heterogeneous for that. And it's really becomes difficult when you have gaps in sample collection or in sample types that you wanted to collect. So then the next, for processing, you want to look at what processing you want to do and how you can sort of future-proof your assays. There's different ways of processing, different ways to store samples. Do you want flash frozen? Do you want frozen in an embedding medium? Do you want to make plasma, serum, buffy coat? Do you want whole blood? Do you want to spin down your urine? Do you want to take your urine as is? So it becomes, again, a question that you need to look at the assays that you currently have intended for your samples and what you think might be useful in the future. I'll give some examples of that later on. The other question is, do you have storage space? Given the relative scarcity of space at the RI, and in fact, in almost all research institutions, finding space becomes an issue for current needs, and also you need to think about future needs, because believe it or not, biobanking, the number of samples can quickly become almost overwhelming. How reliable is your storage system? You have a -80, that's great. Do you have alarms on it? Do you have remote monitoring? These things only fail at like 11 o'clock on a Friday night. There won't be anybody looking at that for 12 hours. If you don't have an alarm, you might lose your samples. And if something does happen to your system, is there a backup solution available somewhere in a reasonable, within a reasonable distance that can be used? And who has the key to the backup freezer is something that really needs to be addressed before the emergency actually arises. And the other thing is that if you start banking a cohort, let's say you want to look at, 3-year survival, this takes a lot of time. It takes, in fact, 3 years to be able to get the data for the samples you've started collecting today. So one thing to look at is whether you want to consider retrospective versus prospective. Well, we'll come to that in a little bit. So the next step after you've decided to there we are. The retrospective versus prospective. In prospective setting, so in other words, you start your banking, and it's the samples that you start banking now that you'll use as your samples for your research. You can precisely define which target population, which eligibility criteria there are for that. You can choose the optimal sample collection method. You can choose how to process it in the way that is ideal for your assays and your project. if you use retrospective samples, so samples that already have been collected by somebody else or have been collected in a clinical setting, you're depending on the population that was selected then, on the methods by which they were processed. But you can get follow-up info now. If you take samples that were collected 3 years ago, you can get 3-year follow-up versus having to wait 3 years in order to be able to look at whether your feature of interest affects 3-year survival, for example. So generally, people try to combine the two approaches to look at available retrospective populations. This can even include public databases, and use those to define target populations for in a prospective setting. So once you've decided to start biobanking and have established some of these criteria, then comes the next step is the application to the Research Ethics Board, because bio and data banks are considered to be a different entity from research projects if you're applying to the REB. So a quick and easy definition is if you're collecting samples that you're going to use up during the study, or you're going to throw them away when you're finished with the study, it's not a biobank, it's a project. If you're collecting and storing samples for future projects that you haven't submitted yet, or if you want to keep your leftover samples at the end of a project for future research, then you're in the biobank setting. And for a biobank, there's the key documents include the framework or the SOPs, informed consent forms, although you also need those for a study. Informed consent forms for a biobank tend to be somewhat different. Recruitment and advertising materials, if applicable, and templates for all of these are available on the REB website. And if the latest version isn't up on the website, it's a good idea to ask, because sometimes the website can be a little bit slow in updating into the latest versions. So identifying a target population, and this is more in the context of preparing an REB application. Most biobanks, especially at the RIMUHC and at the MUHC, tend to be PI-led and to focus on a specific disease or a specific anatomical site. Again, if normal controls are needed, these need to be defined and described. Generally, the recruitment of normal controls is going to be a different process from looking at participants with a specific disease, because patients without that disease, for example, would not be seen in a clinic specific to the disease of interest. It's important to define these populations up front, because if you didn't include them as a target population, if you want to start adding them later on, you go through the whole amendment and justification process, which can take a fair amount of time because the REB is very busy. So informed consent, you need to identify an appropriate time and setting to be able to approach potential participants at a time when they're not, at the time of diagnosis, especially for more serious illnesses, is not a good time. And you need to be able to give them enough time to actually reflect, think about it, talk to family members if they would like to do so, and to be able to come back with questions. Consent should be phrased as broadly as possible within reason, because what the research modalities that are used now, we don't know what's going to be used 5 years from now. So if you specify only a specific set of assays, you may essentially have excluded something that gets developed 2 years later, because the informed consent is very specific to, if you've restricted your options in the informed consent form, you are restricted to what you've said there. So broad language tends to be tends to allow for more flexibility. Of course, it has to be in lay language. This isn't the lay language of lay abstracts, because lay abstracts tend to be meant for members of committees that have spent a fair amount of time looking at grant submissions. Lay language, we're talking, it should be understandable to somebody who left school 55 years ago in grade 4. So it has to be very simple, because otherwise the informed element of consent is not present. You need to define what types of research samples can be used for. Again, if you say it can only be used for research into one specific disease, then the REB will not permit you to use it for use these samples for research into another disease, even if it's related. And there's often the possibility of opting in or out of specific types of sample collection. For example, some patients may say, yes, you can take a tissue sample from surgery, but you can't have a blood sample. So this is something to keep track of and to actually give patients as much freedom as possible to decide how they would like to participate. Need to define the type, amount, and timing of biospecimens collected, and whether or not, if this occurs in conjunction with clinical sample collections, this will minimize the risk and inconvenience for participants, because every time you stick a needle into a patient to get a blood sample, you're incurring a risk. If the risk is being undergone as part of standard of care, you're not adding to risk by taking 10 mils of additional blood at that time. If you're sticking a needle into a patient to take blood for research only, that risk is not part of their clinical standard of care, and it can be perceived as adding to the patient hazard. You want to cover as many eventualities as possible, and it's a good idea to include asking for consent for access to clinical archival samples if those are applicable in your setting. And the point of that will become apparent later. So aside from, there are regular types of samples. Tissue can be from biopsy, can be from surgery. This generally has to go through the pathology department because it's obviously the tissue is being taken usually for diagnostic use, and interference with clinical care is something that we absolutely cannot have happen. Tissue can be processed for various derivatives. You can just freeze it down. You can freeze it in FBS-DMSO to retain viable cells. You can make cell lines. You can make patient-derived xenografts. Just a note, if you make patient-derived xenografts, it's important. You are likely going to be asked to include this in the consent form, because there is a subset of patients who have moral objections to animal research, so may not be willing to allow their samples to be used in animal models. And of course, tissue can be processed to isolate different cell types. Blood, again, there's different ways to collect it, different ways to process it, and depending on your anticipated assays, this is where you would want to look at the collection volumes, what tube you're using, whether you're taking multiple samples or just one time, and how you're going to process it. There are other types of samples that can also be stored. For example, nowadays microbiome is a very popular area of research. You can collect skin swabs, nasal swabs, stool samples. There are other biospecimen types such as urine samples, hair. For each of these, you have to look at the logistics of the infrastructure and whether or not you'll have personnel available to collect and process each of these. For example, a urine sample is something that you can get from a patient when they come for a clinical visit. Generally, it's a produce-on-demand sample. If it's a stool sample, you're generally giving them a collection kit and asking them to bring it back with contents at their next clinical visit. So it's something you have to keep in mind, that you need to have a way to follow up when their next visit will be and to have somebody to go pick up their samples. For processing, consistency is key to be able to compare between samples. So having standardized SOPs with records of the time between receipt of a sample and processing of a sample is key. You will need to be able to have a log for deviations. And the output of your processing has to be suitable for your downstream assay. Just an example, in the old days, plasma preparation included a single centrifugation spin. Take the upper phase, store it, okay, you've got plasma. Then we came up with cell-free DNA. It became an area of interest. Unfortunately, a single spin, usually done at about 1800 Gs, leaves cells in the supernatant, which lyse when you freeze down that upper phase sample and interfere significantly with cell-free DNA assays. So you need to do a second centrifugation of the supernatant of your plasma to remove this contaminant as much as possible for those assays, and which leads to the issue that if you look at long long-standing collections of plasma and you want to do cell-free DNA assays, you probably won't be able to do it just because they were not processed in a way suitable for that format, although it was suitable for all the other assays envisaged at that time. Storage generally goes into tubes. There's a preference for barcoded tubes because those tend to be much easier to keep track of later on, especially when you're 5 years later, you're looking at a tube and wondering, I can't read John's handwriting. Is that a 1, a 9, or a 7? If it's a barcoded tube, you can read the barcode. You should be able to go back to your database and go, oh yeah, that was from the 19th, not the 17th. The types will vary by specimen, depending on the volume that you, of the specimen that you'll wind up with. And one thing to keep in mind up front is how many aliquots do you want to divide your sample into? The more aliquots you have, the more the tubes will cost you, the more space you'll need. Less aliquots you have, the more times you're going to have to go through freeze-thaw cycles if you have multiple assays that you wish to conduct on your sample. So for usually many sample types, or almost most sample types, are stored at -80 or in liquid nitrogen. Having monitored storage, as I mentioned, with backups and emergency procedures is key. And I can't emphasize enough that however much space you have, you're going to probably wind up needing more. So plan for the future, because if there's no space for another -80 and there's more samples piling up, it's going to become an emergency. Record keeping, it's critical to keep an accurate inventory of what samples you've collected, what aliquots you've made, what you've used for an assay, who you gave samples to, or within your own lab, who's holding the sample for assay at the moment, when they came back, where are they right now, and any deviations from storage conditions. Did your -80 go to -20 at some point, and the subset of your samples has therefore been exposed to a different condition from the ones in the other -80? And freeze-thaw cycles, obviously, if you're doing aliquots, because you don't want to compare one freeze-thaw to 4 freeze-thaws. Inventory control is often done through software, the specialized software packages, such as ATIM, which stands for Advanced Tissue Management. This is specialized software for biobanking. It's developed through the Canadian Tissue Repository Network, or CTRNet, which is actually the sort of National Standards Association for Biobanking, which combines both information management system and clinical annotation modules. There are also other options. People have used REDCap. There are some groups still using Excel, although I don't really recommend that. All of these software packages tend to have been built either as clinical annotation with biobanking added on later, or biobanking with clinical annotation added on later. So they each have their own strengths and weaknesses, mainly based on their original purpose. So clinical annotation, so the data part of bio and data banking, it's key to define your required clinical variables and your potential values or definitions up front, because if you collect this data at the time that you get the samples, and 3 years later realize, oh, I needed to collect this other piece of information, going back across, once the cohorts become large, is quite laborious. The other question that you need to ask is whether variables can be directly obtained. Is it an OASIS? Is it something as simple as, BMI today, which is sitting up there on the top line? Or is it something you need to generate by interpretation of primary data? In the oncology setting, do you need staging? Staging is sometimes available for some samples, or for some cases, it's not always available. It can be generated from the existing data, but that requires a significant amount of time and expertise. And of course, double checking entries is strongly recommended, because no matter how good somebody is, there are always going to be some mistakes. And in fact, even in the medical record system, there are occasional errors. Regular follow-up is usually required. As cohorts become bigger over time, this becomes a major burden on staff. If you have 20 patients and you need to follow them up every 6 months, not such a big deal. This is relatively easy. Once you hit 2,000 or 3,000 patients, doing follow-up becomes a major issue. Need to define how often you need follow-up. Do all the variables need to be checked at the same frequency, or there are only a few that you expect to change quickly over time? And will future data will be available within the local record system if you're depending on MUHC records? Because we're, as a tertiary care center, in many cases, we are not the hospital that patients will always be coming back to. So you, in many cases, you'll get, the single event for which they were referred to the MUHC, but then their follow-up can wind up being at their hospital closer to their original residence. There are ways to get data from external sources, such as the Dossier Santé Québec, but there is a significant lag time, and there's also expenses associated with obtaining that data, so that's something to keep in mind. And also the amount of data available is increasing, and the criteria for reported results change over time. For example, if you look at breast cancer, ER positive versus negative, the percentage of cells positive to be able to call something positive has gone from 30% to 10% to 1% over time. So if the only thing you record is ER positivity status, you're probably not recording the same information depending on the time point at which that patient was actually seen. Just an example, 2009, that's a lung cancer pathology report. Simple, there's not a lot of variables you can get from that. 2019, this is what it turned into in a synoptic format. So there's a lot more information being recorded, although mind you, some of it here is just repeating the test parameters. But as time goes on, more variables wind up being looked at. And if you look at your initial samples, those tests will not be redone in those. So that may be an issue with doing comparisons within a cohort. So sources of clinical information. Looking up things manually is time consuming and error prone, but often it's quite unavoidable. For people familiar with OASIS, our clinical database here at the MUHC, it is not good for doing searches. It is not intended for automated data collection across cases. It is meant for a physician treating a single patient to be able to have access to the information for that patient at that time. The information is not always present in consistent locations if you're looking at it from a research data collection point of view. And some data is not going to be present in a simple format. It's going to be in handwritten clinical notes, which are scanned to PDF and included in OASIS. So there is no simple way to extract that aside from going through them and reading them. And as I mentioned before, some information is not present and has to be extrapolated from the data available. There are other data sources, which are what feeds into OASIS, because OASIS is not a database in itself. It's simply an interface to a collection of individual databases, 20 or more across the MUHC. So their individual departments have their own record systems, for example, Cerner and Pathology, which tend to be more searchable, but that access has to be negotiated with the department in question. There is a tumor registry for people working in the oncology setting. This is curated data with a team of dedicated registrars. It's not real time because it takes a certain amount of time for data to be entered into that system. And you need to get permission from the MSSS to access this because this is considered a ministry level resource. It's also the Data Warehouse Initiative, which is essentially an attempt to take the databases that feed into OASIS and make them feed into something that's a more searchable and accessible database for data exports. Addition of data sources across the various databases at the MUHC and extension to previous time periods is an ongoing process. Although there is no direct interface to Data Warehouse, you can submit requests to Data Warehouse for data collected within it, and you'll essentially get digested data back out. And due to obviously time and personnel restraints, very wide-ranging data requests are generally discouraged. The request is that you ask for a specific subset of information on a specific subset of patients, preferably previously consented. So there are other clinical services which can be key partners in a biobanking setting. For example, pathology. If you're looking for tissue, essentially all tissue samples that might be related to a disease state have to be screened by a pathologist prior to release for research use. Also for if you want to be assessing tissue samples later on or constructing tissue microarrays for multiplex IHC or immunofluorescence, your pathologists are your best friends, and this is generally done in a collaborative setting. Surgery is also a service. It's important to be able to interact with to get identification of eligible patients who are going to undergo surgery. Medical oncology, you can see obviously I have my previous experience is somewhat biased towards the oncology setting, to look at interpretation of the neoadjuvant and adjuvant therapies that patients may undergo. Anesthesiology, believe it or not, it sounds like it wouldn't be a service you'd be dealing with in a biobanking setting, but if you're in an operating room and you want to get blood samples, these are the people who are going to get it for you, so it behooves you to be on their good side. Interpretation of imaging data is something that in radiology is a key partner for. There are a lot of other clinical services at the MUHC, depending on the nature of what samples you want and what information you'd like to get. For example, phlebotomy, the test center, central labs. There's a lot of assays that central labs can do on a contract basis. And it's instead of developing the expertise in lab, it's often easier to get central labs to do it. And I can't forget, one of the most important parts is having a good relationship with the nursing and technical staff of the hospital, because these are the people who will be essentially your best friends in interacting with patients, in obtaining samples, in basically integrating yourself into a clinical setting. Just to note that research can't interfere with clinical care or any other MUHC activity. Clinical care is the absolute first priority, and research is, only after all clinical care activities have been successfully performed. Other potential synergies are, for example, clinical pathology. Believe it or not, they are actually the largest existing biobank at the MUHC. They process, annotate, and archive in a very organized way over 70,000 samples per year. And if you include access to pathology archive in your consent form and you get the patient's consent, a lot of the samples here, after they've been used for the required assays for clinical care, can be accessed for research purposes, of course, with approval for pathology. And the pathology department, in fact, has a formal process for sample retrieval. There's a fair amount of paperwork involved. The cost is quite reasonable. So just a brief summary slide, just to show what the various elements of biobanking are. Goes all the way from identify your eligible participant up front. Just to note that if you want to identify these from medical records, you need specific permission from the DPS, because you would be accessing medical records before you're asking for consent. So that's something that requires a specific approval. Then request consent, get your sample, collect your sample, process it, record the processing data, put it away, make sure that it goes into the inventory control. Then it can be used for research. Associated with research data, there's follow-up, the clinical information and follow-up. Clinical information goes into your data bank as well. And then the key part is this correlation between research data and clinical information and the analysis thereof. And with that, I hope people are still relatively awake, and open the floor for questions and any potential discussion. Thank you for your time.
Thanks, Nick. That was a very helpful overview. Maybe I'll well, there's a question from Stephanie. She's asking in the chat whether you have access to DSQ for research. I guess I would, well, I'll let you answer that question.
Well, it depends. Okay, so it depends partly on having thought up front to include access to DSQ in your consent form. Because if you've written only that the patient's consent for access to their local medical records, there's going to be a bit of an issue with submitting a request to DSQ. But yes, requests can be sent to DSQ for research purposes. However, there is an associated cost. I don't remember the exact amount at the moment, but it's not particularly low. And from what I've heard, it can take 6 months to a year sometimes to actually get information back, because the questions that you're asking are research questions and not the clinical information that's the sort of the main point of the DSQ.
Okay, thanks. I have a couple of questions. So the first, I guess, is, so my personal research interests, I think, would benefit from, I guess, building or accessing a biobank. My first question is, what are some examples within the RI where you have sort of witnessed like a well-functioning and actually used biobank? I know that you're involved in the lung tissue or lung cancer space, but can you just give us an example of one that's sort of, one, accumulating specimens in, and two, being used to answer research questions, and maybe speak to whether it's being used sort of by PIs at the RI, or is it being used by industry, or both?
Okay, so first of all, you have to add a caveat to that, is that there are many biobanks at the RI, and the biobank platform is essentially a contract platform which manages a subset of those to different extents. So essentially we offer services. In some cases, we're doing pretty much all the services. In other cases, for some biobanks, we're doing only collection or only processing. And for other biobanks, they have it's all of their own staff doing their work. So I would not be able to speak to any of them. So for anyone who's from one of the biobanks that we don't have significant interactions with, my apologies in advance for not citing you at this as an example. For example, if you look at, there's probably one of the longest standing examples is the breast cancer biobank, which was founded back in 1999, and data from which, or samples from which, have been used to support a pretty wide variety of studies and publications, and used to as a background for grant applications. These, in general, in my experience, these biobanks tend to be used primarily by PIs across McGill, the RI, the MUHC, and often wind up associated with other biobanks across sort of the local community, either in Quebec or Quebec and Ontario and the Maritimes, or even at the national level, to create larger biobanks, because of course the bigger scale you have, especially when you're looking at rare subtypes, the more you have in the way of statistical power. Looking in the commercial setting, this tends to be a little bit difficult, especially Quebec's sort of legal and ethical framework is a bit restrictive on the viewpoint of selling samples, and there's clinical trials are, in a way, they're biobanking, also a special instance of biobanking, in that pharmas will get the tissue themselves, but generally they also legally restrict that you can't research biobank the clinical trials cases, so that you can't scoop their data. There has been a certain amount of interest, but as I said, there legally and ethically it becomes a bit of an issue. There is, of course, the option of participating with industry in industry-funded collaborations, for example, in the context of CQDN grants is there, but in the handing tissues directly over to Pfizer, for example, this has been quite rare.
Okay, that's helpful. There's a question and a comment in the chat. So Severin asks if you have information sort of on the expiration for various types of samples? For example, how long would human plasma be able to be kept at -80?
It depends completely on the assay you're using, because obviously some analytes are going to be more stable than others. I know that looking at RNA from tissue, we've looked at RNA from tissue that has been stored for 15 years, and it's still pretty good. The question is, of course, if your freezer failed at any time in those 15 years, you probably cooked all of it. But it's very dependent on your storage conditions and on what you're looking at. I mean, I wouldn't be looking at maybe cell-free RNA in 15-year-old plasma.
My question was more like, is there any standard in terms of, or how long we keep samples, because we know they are no longer good after a certain time? Or you kind of answered this question saying like, well, it really depends what we want to measure, right?
And in a lot of cases, nobody's actually done the assay because nobody's wanted to wait 25 years to see whether their stored plasma still works.
Yeah, it's just like, because you mentioned it can become very expensive to keep samples for large amount of samples. If we keep samples forever and ever, and we don't know if they're any longer good.
So what I would suggest in that case, if there's a particular assay, the suitability of which you want to look at is, make multiple aliquots of one control sample that you have a lot of, and pull it out every year and run the assay on it. When you see your values start to suddenly do this, you know that your samples aren't good for that assay anymore.
Yeah, I was just okay. Thank you.
The problem is, I mean, I hate to answer a question with "it depends," but in this case, it really does depend. I mean, a frozen tissue sample, you probably use it for immunohistochemistry. Pathology department has samples in beeswax from the 1930s somewhere. They probably still work for immunohistochemistry. Not perfectly, but some of them will.
Yeah, well, may not. Yeah, okay, thank you.
Depends on the antibody.
So Ayat Salman points us to the liver disease biobank, which has been around for a decade, as a successful example. That's helpful. So my last question, maybe, and then we'll wrap up unless anyone else has a question, is probably the stickiest one. Is it's not clear to me the funding model, where a lot of grants that I apply to will explicitly say that the funds can't be used to support a biobank. They want it all to be, hypotheses that will be tested now. And so they encourage you to, you can access existing biobanks and there's tremendous value in that, but you cannot kind of be contributing to biobanks. So where do the biobanks that you've worked with, or the platform, and maybe this liver disease biobank, for examples, but like, how are they funded? Through foundations, through just mystery sources? Like, what's the model there?
There are some mechanisms for specifically biobank funding. Mm-hmm. There's the Réseau de Recherche, for example, again, looking at the oncology setting, there's the Réseau de Recherche sur le Cancer of the FRSQ, which has a biobank, bio and data banking axis, which does have some funds available, although they're relatively restricted. The problem with biobanks is essentially you wind up maintaining them, unless you're using foundation funding, which tends to be sort of a more long-term, you're maintaining them with, a bit from this grant, a bit from that grant. 50, you've dedicated 25% of a research assistant on this grant to data collection, and that's your data collector for the bank for the term of that grant. You write for another one in the next next round. It's like CFI, where we don't have an ongoing source of funding for resources. You're basically scrounging and you're looking at rounds of funding. For example, there was a lot of money went into a Marathon of Hope from Terry Fox. And that, again, depends on accessing, the stuff that's in existing biobanks, but there's no funding to really build biobanks from that. So it's like trying to, get money for a service contract on your equipment.
Yeah. The the respiratory research network in Quebec, there's also sort of opportunities specifically, or there's funding for a biobank, but it's like you said, it's hard, and to me it's unfortunate because the return on investment over time becomes huge because of these really unique specimens. So okay, well, I guess we'll end there. I don't see other questions, but this has been actually very informative to me. So thank you very much, Nick. I hope others also found it helpful.
No problem at all. And I'd be very happy to answer questions of people, the biobank platforms on the web, and my contact information is there. I'm always happy to answer questions. And yes, looking at the chats, yes, this Dr. Matrakis's biobank was one of the ones where I said, I apologize in advance if I don't cite people, because I don't want to steal anybody else's thunder. So.
Okay, you're also getting praise as well from Elena and others. So thanks so much. Have a great day, everyone.